So this is lecture 4 of Principles of Metabolism, and in this lecture we'll start looking at some actual specific pathways. It's been a lot of general concepts so far. And we'll start looking at carbohydrates and some of the main pathways for processing carbohydrates. So what's a carbohydrate? Well, it's literally carbon with water. So it's generally compounds that have this type of a sum formula. It has a number of carbons and it has a number of water constituents in it. The exact definition is a little unclear sometimes, but roughly this is what it is. And carbohydrates are, of course, the primary product of photosynthesis. Plants make sugar generally, and this is why it has a central role in metabolism and in biology. Glucose is certainly one of the most well-known carbohydrates, well-known sugars. It looks like this. One thing to note is that glucose and many other sugars have this property that they can exist both in a ring form, like this, and in a linear, sort of open form, like this. And these two forms spontaneously interconvert. The ring form is much more common, so you typically see structures drawn like this. But sometimes it's useful to look at the open form because it makes the functional groups a bit more clear. And this can be important when trying to understand their metabolism. So glucose is an aldehyde sugar or an aldose. And that's because it has a group like this. It has an aldehyde group towards the end. These sugars are a bit more reactive than some others, but it's a particular class of sugars. Other examples of this group is glyceraldehyde, which is perhaps, glyceraldehyde, which is perhaps even more reactive than glucose and a bit toxic in high concentrations. Ribose is also an example of an aldose sugar. Then there are the ketoses. So this is another type of sugar. So these differ a little bit in that they don't have the aldehyde group, but they have these type of ketone groups instead. So fructose is one example, another major sugar in human metabolism. And dihydroxyacetone is also an example of one of these sugars. So you can tell sometimes the names are really not very helpful. So dihydroxyacetone, that really doesn't sound very tasty. It doesn't sound like a sugar, but it is. So it's all it means is that you have this ketone group here, and that's an acetone group, you might say. So that's also sugars. Then there are the alcohol sugars. So these are maybe less common in human metabolism, but the intermediates in pathways. Sorbitol, I'm sure people have heard of, so it looks like this. And now you have an alcohol group instead of a ketone group as in fructose. So alcohol sugars. Sometimes these are called polyols, so lots of alcohol groups. And so you see these in metabolic pathways. Glycerol is another fairly common alcohol sugar. Exists as the backbone in fats. So that's carbohydrates. Now, carbohydrates exist in this open form or in this free form in the metabolic pathways we'll look at. But of course, there are also complex carbohydrates. We're not going to talk about this so much, but this is usually the storage form, glycogen in humans and other complex carbohydrates in other species. Okay, so that's carbohydrates. Now, what can you do with them? So one of the reasons that carbohydrates are very important is because of the glycolysis pathway. So this is a bit of a unique pathway because sugars are really the only substrate where you can get ATP without using oxygen. So this is a pathway that can extract energy from carbohydrates without any net oxidation. And that is unique to sugars, actually. That's important to look at. So how does this work? So glycolysis is usually divided into two parts, and usually it's drawn top to bottom. So you have an upper part, upper glycolysis, and you have something called lower glycolysis. So in the upper part of glycolysis, what happens is that glucose, which is a hexose, a 6-carbon sugar, first gets phosphorylated twice. So it's actually using 2 ATP to do this. So this is energetically costly. And then there's also a bit of isomerization, so the glucose actually changes into fructose. and then the fructose has two phosphate groups added to it by the ATP. So you have this doubly phosphorylated form of fructose. And then comes the cleavage step. So glycolysis is actually the cleavage of sugar. So this 6-carbon sugar gets cleaved into 2 halves. So you have 2 times this sugar, a glyceraldehyde phosphate. So an aldose sugar with a phosphate group on it. So that's sort of the preparatory step. And so far we have actually expended energy in the form of ATP. But then comes lower glycolysis, and now we're at the payback step, so to say. So this is the steps that are able to generate ATP without having a net oxidation. So this is really the unique part, I think. So how does this work? So briefly, as an overview, this is using glyceraldehyde phosphate produced in the upper part. And then there are several steps between this compound and pyruvate, and we'll look at this in more detail shortly. And this is the oxidation part, you might say. So actually, there is some redox going on here. There is oxidation going on. And here you get 2 ATP out of this process for each glyceraldehyde phosphate. But remember, you had 2 of them here. So from a glucose, you get 4 ATP in this part. Whereas you had a cost of 2 ATP here. So net, you will have a gain of a total of 2 ATP from this whole system. So this is an oxidation step. You can see that there is an NAD here also that gets reduced. So it's accepting electrons. And then you get to pyruvate. But then there is a final step where the NAD can be reduced back from NADH to NAD to produce lactate. And so you see that this is a closed loop. This NAD is recovered again, and the electrons actually come back to lactate. And so there is no net oxidation here. So you have, as a total, gained 2 ATP from the glycolysis of glucose all the way to lactate. And there has been no net oxidation. So that's kind of unique. So a bit more in detail, how is this possible? Because usually we expect that oxygen has to be involved for this to work. So here's what's going on really. Really what occurs in lower glycolysis is a sort of clever rearrangement of electrons in a way so that you have a more favorable compound towards the end. And that's how you can gain some energy. So the key reaction, I think, is the glyceraldehyde phosphate dehydrogenase. So this is an enzyme that takes glyceraldehyde. It's a dehydrogenase, which means that this is a redox enzyme. And it's being a bit clever. So it's using the fact that we have a reactive group on glyceraldehyde, an aldehyde group. And this aldehyde group can capture a phosphate and add it onto here. And it can do that without using ATP. So this is not a kinase. So it can just capture a phosphate. And in the process of doing this, it's also doing a redox reaction. Because whereas we used to have a hydrogen here, we now have an oxygen. And that's a much better deal for this carbon atom here. So this reaction is also able to extract a couple of electrons and reduce NAD to NADH. So this is a good deal. So we get to phosphorylate this compound without using ATP, and we gain 1 NADH in the process. And NADH is an energy-rich molecule. So in the next step, now we have a doubly phosphorylated glycerate. This is also a high-energy compound. And this can be used to phosphorylate ADP to ATP in itself. So one of these phosphate groups, this one, is added onto ADP to phosphorylate it. And now you have this thing left. So here's one of the ATP gains. And then there's a couple of steps. There's an isomerization, there's a dehydration or a removal of water. And I'm not looking at those in detail here. But after those steps, you have rearranged the structure a little bit. And now you have something called phosphoenol pyruvate. And this also has a reactive group. So it has what is known as a phosphoenol group. It's a pretty rare functional group in metabolism, but it occurs here. And this is, again, reactive enough so that this phosphate can leave and phosphorylate ADP on its own and make ATP. So this is the pyruvate kinase reaction. So this is a kinase. If you would think of it in the opposite direction, it's phosphorylating pyruvate, but actually the reaction is favorable in the left to right direction because phosphoenol pyruvate is actually higher energy than ATP itself. So this is what's going on, and in this way the lower glycolysis is able to phosphorylate 2 ADPs into 2 ATPs using taking advantage of the structure of these sugars. So we mentioned that there is actually redox going on. There is NAD involved in glycolysis. So in the first steps of lower glycolysis, where you gain the ATP, you have an NAD being reduced to NADH. And then in the step between pyruvate and lactate, this goes back again and sort of closes the circle. And this is what has to happen if you have no oxygen, because the electron has to go somewhere. And the only way to figure this out for the cell is to regenerate NADH this way. And that generates lactate, and then you can't really do anything else with the lactate. So this just gets spits out. And this is the amount of ATP you can extract from sugar if you don't have oxygen. Usually we do have oxygen though, and glycolysis of course works to catabolize sugar also when you do have oxygen. And in this case, first of all, the pyruvate usually goes into the mitochondria, and so you can oxidize that, and you can get a lot of energy out of that. And we'll look at this later. But in this context, it's also important to remember that the NADH that is made in lower glycolysis is actually very energy rich in itself. So in addition to the 2 ATP you get here, this NADH can shuttle into the mitochondria. And here an NADH is worth enough to make 2 or up to 3 ATP. It depends a bit on how the respiratory chain is working. So actually, in the presence of oxygen, the glycolysis process is able to extract the energy worth of, say, 4 ATP, 2 plus 2, or maybe almost 5. So this is an important piece of energy extracted from sugar as well, which is sometimes forgotten. So that's important to know. So one problem with glycolysis, which is talked about a lot, is acidification. So glycolytic metabolism is known to be acidifying. So it lowers pH, it produces acid. And why does this happen? So it's a common conception that glycolysis produces lactic acid. That's actually not entirely true. So glycolysis produces lactate. And lactate in itself is a base. So this is not acidifying. If anything, it makes the pH a bit more alkaline. But there is a problem with this mode of metabolism, because the ATP that you produce in glycolysis, if the cell is relying entirely on this in anaerobic metabolism, has to be used. And when ATP is hydrolyzed in any reaction that requires ATP, like a proton pump or protein synthesis or something like that, it generally produces 2 protons. So this is the acidifying part. So if you couple glycolysis with ATP hydrolysis and you have no respiration or anything like that going on, then the net result is that glucose gets converted to lactate. You do a lot of work with the ATP that you make, but you get a leftover product of 2 protons. And this is why glycolytic metabolism is acidifying as a total. If you do respiration, then it turns out that the respiratory chain can capture the protons that come from ATP hydrolysis. And so when you have oxygen available, metabolism is pH neutral. This has of course been ensured by evolution, because producing acid is not a good thing for an organism. But when it comes to anaerobic metabolism, there is really not much of a choice. So this is what happens. This is detailed in a great article by Peter Hoshachka in Science 30 years ago now. But it's a good article to read if you're interested in this kind of stuff. One also important point with this is that lactate dehydrogenase, so far from actually producing lactic acid, it actually helps. in this setting when you're doing anaerobic metabolism. Not only is it required to regenerate the NAD that you have to have if you're going to run glycolysis in anaerobic mode, but it also actually captures one proton in this process. So lactate dehydrogenase is a critical enzyme in anaerobic metabolism because of this. And so if you don't know about this, you might think that blocking lactate dehydrogenase would help with the acidification, but it's actually quite the other way around. If you block this reaction, you will have lots of problems. So that's about pH and acid metabolism in glycolysis. So another interesting aspect of glycolysis, and this applies to many parts of metabolism, I think, is the importance of phosphate groups. So you notice that glucose is immediately phosphorylated when it is taken up into the cell. This is the first reaction, hexokinase. You put on a phosphate group. And if you look at metabolism, to go through these steps of glycolysis, you'll see these phosphate groups are on every intermediate. It's always there until you get to pyruvate. Then it's cleaved off in the last ATP generating reaction, and pyruvate is then free of phosphate again. But everything else stays phosphorylated. And this is important for the energetics and for making ATP and so on. Those are phosphorylating reactions. But there's another interesting part here, and that's that phosphate groups help to trap metabolites in the cell. So there are carriers that take up glucose into the cell, but once you have phosphorylated glucose, it no longer fits into those carriers. So this is also a way of trapping glucose and concentrating it and making sure that none of these intermediates escape out of the cell. So that's an important aspect of the phosphate groups. So all in all, glycolysis is of course a favorable process. It occurs spontaneously. This is how we can extract ATP from sugar in this way. And because this is favorable, going from glucose to pyruvate, it cannot just be reversed if you want to synthesize sugar. And human cells have to do this sometimes. So hepatocytes notably synthesize a lot of sugar in gluconeogenesis, so the resynthesis of glucose. So how do we do this then? Because we can't just go back up the pathway. So there's a couple of enzyme systems that bypass a couple of steps in glycolysis in order to be able to reverse it. And the first step that has to be bypassed is the pyruvate kinase step, which is down here. So this is a step that is irreversible. It uses the high energy compound phosphoenol pyruvate to make pyruvate. So we have to come up with something else if we're gonna get back to phosphoenol pyruvate. And gluconeogenesis can start in with different compounds. It is commonly made from lactate. That would be the core recycle in human metabolism, where lactate is converted back to glucose. And then glucose can be used in anaerobic metabolism in other tissues. So if you start with lactate, you first have lactate dehydrogenase. This is this step. And then you have pyruvate. I'm not showing it here, but that's the intermediate. And then you have an ATP requiring step. So here's where you have to put in extra energy to be able to reverse this. So you have an enzyme known as pyruvate carboxylase, which can add a carbon dioxide group onto pyruvate to make oxaloacetate. So this is an organic acid, and it basically differs from pyruvate by having this sort of carbon dioxide group tacked on here. Oxaloacetate can also be taken from the TCA cycle, and it can be generated from amino acids. So these can also be substrates for gluconeogenesis. And once you have oxaloacetate, you need another step. So this is what is known as a carboxykinase. So this is a clever enzyme that takes away this carbon dioxide group again, which is a favorable process, and also puts in a bit of extra energy in terms of a GTP at this time that is hydrolyzed, so that you get a large energy push and is able to recreate this energy-rich compound, the phosphoenol pyruvate. Once you've done that, a lot of the reactions in glycolysis are pretty much reversible. So it turns out that you can go all the way up to here, up to the doubly phosphorylated fructose, the biphosphate. That's okay, that can happen on its own. But then you need to bypass a couple of steps. So this step, going in the glycolytic direction, is a kinase, and we need to bypass that step. So we have a phosphatase, or a biphosphatase, that just cleaves off one of these groups, and for that you don't need any extra energy, but you use the phosphate group in fructose bisphosphate to do this. But you need a separate enzyme because the kinase would not be able to do this. The kinase that you have here would have to produce ATP if it would reverse, and that can't happen. And then the next step is an isomerization. So this is okay to reverse. There's no energy really involved there. And then in the last step, again, you have to remove a phosphate. And again, this is a kinase. This is hexokinase going in the glycolytic direction, and that's not reversible. You can't use that to go backwards and make ATP. So you have to have a phosphatase again. So there's an enzyme, a phosphatase, that just cleaves off that phosphate group, and then you're back to glucose again. So in order to actually synthesize sugar, we have to bypass a couple of steps. And we do that by inputting energy down here in the form of ATP and in the form of GTP. And we do that by foregoing these kinase steps and instead using phosphatases that don't give ATP in this case. So there will be a net cost, an additional cost of ATP and GTP in order to make this happen. So the final mode of metabolism we'll discuss with carbohydrates and glucose is a part of the pentose phosphate pathway. So while glycolysis uses glucose, sugar, to make ATP, this pathway uses the sugar to make NADPH. So as we have said before, NADPH is the major electron carrier that is used in reductive metabolism. So it's used for anabolic pathways usually, but it has to come from somewhere. And this is one of the few reactions that are able to generate NADPH so that it can be used in other parts of metabolism. So it's a fairly simple pathway. It starts with phosphorylated glucose, similar as glycolysis does. There's the first oxidation step that takes a couple of electrons from the glucose and adds it onto NADPH. So this is one of the objectives of the pathway. You are able to make one of these energy-rich molecules. Now you get a kind of sugar that is called a lactone. It has to do with, again, with how the functional groups look. It's not super important what it's called, but it's important to see that you have had an alcohol, a hydroxy group here, and now you have this type of a ketone group. Actually, this is the lactone part. So that's an oxidization. You can see that you have a double bond to oxygen. Then there's an isomerization. So this ring is cleaved or opened, and you get something called 6-phosphogluconate, but it's pretty much the same thing. It's just changed a little bit in structure. And then you have the second oxidation step. And this oxidation step actually removes a carbon dioxide. So this part of the molecule, which was kind of exposed by isomerizing it, is now a nice carboxyl group that you can cleave off into CO2. And that's a favorable reaction. So again, we can capture a couple of electrons here and make NADPH. So that's, again, the goal of this pathway. And when we have done that, we have taken off one carbon from the 6-carbon glucose. So now we have something which is 5-carbon. It's a pentose sugar. This particular sugar is ribulose pentose. This is not used for many other things in human metabolism, we think. So it is converted to ribose, which has a lot of usages. It's present in nucleotides and many other compounds. It can also be converted back into glycolytic intermediates. So this pathway is sometimes called a shunt, so that you exit from glycolysis up here, and after a few reactions, this thing can actually go back to glycolysis. That is done through another part of the pentose phosphate system, which we will look at a bit more closely in exercises and in one of the computer labs. But this is the second major usage of glucose to generate NADPH and reducing power.